Chain reaction is a method for producing very large numbers of copies of a specific DNA sequence without having to clone it, a process called amplification. PCR was invented by Kary Mullis, who received a Nobel Prize in 1993. A polymerase chain reaction consists of 5 major components. The first component is the target DNA to be amplified. The next 2 components are chemically synthesized single-stranded oligonucleotides, each of which is complementary to one end of the target DNA. The fourth component is the heat-resistant enzyme taq-DNA polymerase. All of these ingredients are combined in a tube and placed in the final component, a thermocycler, an instrument that is programmed to change incubation temperatures and times. The PCR reaction is actually a series of reaction cycles controlled by the thermocycler. The first cycle begins when the temperature is raised to 95 degrees, causing the strands of target DNA to denature to single strands. Next, the temperature is dropped to between about 37 to 65 degrees, the exact temperature depending on the primers and how well they match the base sequences of the DNA. In this step, the 2 primers anneal or bind to their complementary sequences on the target DNA. Finally, the temperature is raised to 70 to 75 degrees and the taq polymerase extends the primers in the 5' to 3' direction. At the end of this first cycle, there are now 2 copies of each original target DNA. Note that the newly synthesized DNA on one of the copies extends past the primer on the other copy. The second PCR cycle proceeds exactly like the first. The thermocycler raises the temperature to 95 degrees to denature the strands. Then the temperature drops to 37 to 65 degrees to permit the primers to anneal. And finally, the temperature rises to 70 to 75 degrees to permit replication. At the end of the second cycle, there are 4 copies of each original target DNA fragment. Cycle 3 proceeds exactly like the previous 2 cycles. The thermocycler controls the temperature to permit denaturation, primer annealing, and replication. In each cycle, the number of replica copies doubles. At the end of cycle 3, there are now 8 copies of each original target DNA. Note that at the end of this cycle, there are now unit-sized DNAs that extend only from one primer to the other. As the cycles continue, these will become the predominant PCR products. In the next cycle, as in the first 3 cycles, the thermocycler continues to control the denaturation, annealing, and replication steps by repeatedly changing the temperature. At each cycle, the number of DNA copies doubles. By the 10th cycle, there are 1,024 copies of each original target fragment, and by the 20th cycles, there are 1,048,756. The PCR technique has revolutionized biology to at least the same degree as has recombinant DNA techniques. Other examples of the use of PCR include allele-specific oligonucleotide hybridization and DNA typing.